Hello my friends, welcome to another Cyanate video. In this one we're going to be looking at photosynthesis as part of the bioenergetics topic, where we'll also look at respiration in a different video. In this one we're going to look at what photosynthesis is, we'll look at how to do the required practical of investigating the rate of photosynthesis. We'll then look at the factors that affect the rate of photosynthesis and how to think about what the uses of glucose are. Let's get to it. In the AQA course, this is the last topic of the paper 1. We're here looking at photosynthesis rates and use of glucose, and as I say, we'll do a later video on respiration. So, what is photosynthesis? Well, let's look at the word first. Photo, light. Synthesis, making things. So it's using light to make things, and it's an endothermic chemical reaction using light as an energy source to make glucose and oxygen from carbon dioxide and water. Some of this you would have looked at in primary school. I mean, the glucose and oxygen, or sugars and oxygen from carbon dioxide and water using light, that absolutely is covered in primary school. The glucose part, maybe not. That's just the word we use for the sugar. That's the name of the sugar. And endothermic, you probably haven't even seen before unless you've done it in chemistry. But endothermic, if you break this word down, you've got therm, means heat or energy, and endo means into. So what this basically means is it's taking in energy. It's taking in energy. It's a chemical reaction that takes in energy. There are some reactions that are exothermic, they give out energy. This one is an endothermic, it takes energy in. So it's taking energy in, using light as the energy source to make glucose and oxygen from carbon dioxide and water. We've got a word equation which we need to be able to use, which is carbon dioxide plus water goes to make glucose and oxygen. This is a word equation. If you were to write O2 rather than oxygen, you would not get marks. You must say oxygen. And I've also got here that light is added in. It is in this extra part. Light is not a physical part of this equation because it's not a physical thing. It's packets of energy. It's not molecules or atoms. And we also need to know the symbols of these. So carbon dioxide, CO2, water, H2O, glucose is C6, H12, O6, and oxygen is O2. We just need to be able to write their symbols as well as this word equation, but you could write a symbol equation for it too, which would look like that. Now, the structure that's doing the absorbing of light is a molecule, which is found in chloroplasts, which you've looked at in the cell structure, cells topic, and these chloroplasts contain a molecule called chlorophyll. And chlorophyll, it basically is the green part of a plant, and that is the structure that absorbs the light to trigger this whole process. It triggers a reaction between carbon dioxide and water to make the glucose. Now, with that in mind, the factors that are going to affect the rate of photosynthesis are the two main parts of the equation, CO2 and water, although water is very readily available for plants. So we kind of ignore that. So CO2, very important. The higher the CO2, the faster the rate of reaction. So that's CO2 concentration. The higher the light intensity, the faster the rate of photosynthesis. The more chlorophyll that is around in the plant, the more photosynthesis can happen. So if it's got a good source of nutrition, it's able to produce lots of chlorophyll, it's going to photosynthesize faster. And the other part is temperature. So temperature being close to the optimum for its enzymes will encourage photosynthesis to work best. This part is for required practical, so we all have to have done this, and it's about investigating how fast photosynthesis can work and changing a factor that might affect that. Now, we can measure it quite simply by just collecting anything that tells us that photosynthesis is happening, which is either it's producing glucose or it's producing oxygen. Now, it's difficult to collect and quantify glucose, so we're more focused here on the oxygen. And oxygen is obviously a gas, and it's going to be released through the stomata of the plant, and so we can therefore collect it and we can see how much they're producing. We could also see how much the products have decreased. So water is a bit more tricky, but we could see how much the presence of CO2 has decreased, so we could track the concentration of CO2 in a closed chamber and see how that decreases, and how quickly it decreases tells us how quickly photosynthesis is happening. But oxygen here is being produced And so if we use certain setups, we can collect the oxygen and we can see how quickly it's being made and therefore produce a rate. Because remember, a rate takes into consideration the idea of time. And so we're looking at per second or per minute or per hour or per day how much oxygen is being produced. So the most common setup in these required practicals is this one here. So you've got here a pondweed. It could be something like Elodea, which is a genius name, whoever came up with that. Elodea is a really common pondweed, and pondweed, when it's photosynthesizing, will produce oxygen, and it will therefore, because it's in water, it will produce oxygen bubbles. So you could sit there for a minute and you could count how many oxygen bubbles are being produced. And that gives you, therefore, a rate of photosynthesis. Now, of course, it's not very accurate, and getting that to work is almost an impossible task. And if you've managed to have it work in class, well done to you. But the other thing that we could also use is something called a gas syringe. Now, a gas syringe is much more accurate than just counting bubbles because it gives us an absolute volume of the gas that's being produced, and we could leave that for 24 hours. A gas syringe we literally connect to the end of a boiling tube, and it will then be pushed out as oxygen is being produced. And if you use a small enough gas syringe, it'd be nice and accurate. You get a nice measurement from it. And of course, for these, to increase the accuracy, we want to be doing lots of repeats. So you want to be repeating it 3 times or more and calculating then a mean. And that's critical in almost all experiments because it removes the risk of outliers or anomalies, and we can then eliminate those from our results. So how do we set this up? Well, that's the method really. The independent variable for this experiment, as usual, is the one that you're investigating the effect of, the thing that you are changing. In this case, you could change any factor that would affect the rate of photosynthesis. So it might be the light intensity, it might be the concentration of carbon dioxide, it might be the pH of the soil, it might be I don't know, any factor that you wanted to investigate, because we're not talking just here on a small scale. In a lab, you could be talking agriculturally. What process, what factors are going to help photosynthesis to happen the best? The dependent variable is always the thing that you measure. So in this case, we're measuring the rate of photosynthesis, but it's how do you do that? So is it bubbles per minute, or is it volume of oxygen made per second? You're obviously not going to count and take a measurement every second, but you can collect results over 5 minutes and then shorten it down to 1 second. So it's really, really useful to think of it in terms of that. The controlled variables are the things that you're going to then keep the same, and these 3 variables, categories of variables, are always important in experiments. The controlled variables are the things that you keep the same. There's no point running an experiment if you're changing lots and lots of things at the same time. You need to have just one independent variable, one thing that you're changing, one thing that you're measuring. You can measure more, but then the controlled variables must be kept the same. So if you're going to change the light intensity, you must keep everything else about that plant the same. So the same plant, the same type of plant, the same volume of leaf, the same light if you're measuring light intensity, of course you're going to change that, but you can change or keep the same the type of light, the wavelength of light, the color of it, if you like, the lamp that you're using. You need to keep the same the concentration of carbon dioxide, the temperature of the room. Everything else needs to be kept the same, so you're just measuring the effect of changing light intensity on the rate of photosynthesis. As we covered earlier, there are 3 main factors that affect photosynthesis, 1 being light intensity, 2 CO2 concentration. Both of those are in the equation for photosynthesis, therefore they're obvious. Amount of chlorophyll is also very important. The more chlorophyll you have in chloroplasts, well, the more chloroplasts you have, the faster it's going to happen, because you've just got more sites of absorbance of light. And there is an extra factor here, which I haven't put directly because it more affects all rates of reaction, and that's temperature. And temperature is absolutely critical. We've got enzymes here which are controlling the rate of reaction, and if the temperature goes too high, the enzymes will denature. If it goes too low, the rate of reaction will just decrease. So we want to work around the optimum of the plant for the optimum rate of photosynthesis. Now, a limiting factor, this term here, a limiting factor, is the factor that's stopping photosynthesis from getting any faster. So if we've got a really high light intensity, we've got a really high amount of chlorophyll, we've got perfect temperature, but we don't have much CO2, this CO2 is what we call the limiting factor. It's stopping the rate of photosynthesis from increasing anymore. So for example, we could have the rate of photosynthesis. I'm going to put up the y-axis here, and I'm going to put light intensity across the bottom, and this is going to be a graph that shows how light intensity affects the rate of photosynthesis. Now, to begin with, if you increase light intensity from 0 down here, the light intensity increases. It's going to increase the rate of photosynthesis, and it's going to be a proportional relationship. And as you get to a point, if you keep increasing light, the rate of photosynthesis is going to kind of tail and flatten off and plateau and not increase anymore. And that's because you're giving it more and more and more and more light intensity. It's got all the light it could ever need, but there is a different factor which has now become limiting. So perhaps here there's not enough CO2 to keep up the amount of light that's being provided. Perhaps the temperature is not warm enough to encourage the enzymes to work fast enough and keep up with the rate of photosynthesis it needs to match. And you could do exactly the same. You could change it for a CO2 graph with, again, rate of photosynthesis here. If you increase concentration of carbon dioxide, the rate of photosynthesis is going to increase in a positive correlation, proportional kind of way. And then it's going to get to a point where it then flattens out, it plateaus, because something is a limiting factor here. It's not CO2, there's plenty of CO2 at this point. In fact, CO2 is a limiting factor. At this stage here, in this bit, CO2 was the limiting factor, because as we're increasing CO2, as CO2 is going up, the rate of photosynthesis is going up. So therefore, CO2 was the limiting factor. But in this bit, there's something else that's a limiting factor. So if you're a farmer and you're thinking, well, I'm providing all this CO2, it's costing me money in this closed climate, closed area. I'm giving all the CO2 that it needs, but the rate of photosynthesis isn't increasing, the plants aren't growing any quicker. There must be a different factor that's slowing it down. And that would probably be light or temperature or chlorophyll amount, that sort of thing. Now, if you're doing higher tier, it's likely that there are factors that interact, and any one of these other factors might be the limiting factor. So it's quite complicated to think about. But when we're keeping it simple, CO2 was increasing and the rate was increasing, therefore CO2 was the limiting factor. When we're increasing CO2 here and the rate isn't increasing anymore, there must be something else that's stopping photosynthesis from getting any faster. The final part here in limiting factors really is a very high tier concept, which is explaining how light intensity affects photosynthesis by use of something called the inverse square law. And the inverse square law is a way of explaining how light spreads out from a source. So if you've got a light source here, the light will spread out in all directions, but here we're just focusing one direction. And if we imagine a distance of, let's call it 1d away, the light intensity is going to hit this little square, and we'll call the amount of light intensity that hits that 1. If we then move to a distance of 2d, so twice the distance, each one of these squares now gets 1/4 of the original light intensity. And in the next one, each square only gets 1/9 of the light intensity at a distance of 3d. And this is a law called the inverse square law, where the light increases following this pattern. And so the light intensity is actually decreasing in each of these little of the same area as we move further away from the point of origin of the light source. So with this in mind, we can plot a graph of the inverse square of distance away from the light source compared to light intensity. And if we do plot this graph, we get a directly proportional relationship that as the distance increases, the inverse square of distance is decreasing. But as the distance increases, the light intensity will decrease. And if the distance decreases, the light intensity will increase. And the light intensity here can be changed for rate of photosynthesis because they are directly linked together. And this is the relationship that is used to explain and describe the pattern for how light intensity affects photosynthesis. So it's the inverse square law. So if you're asked a question, which pattern describes how light intensity affects photosynthesis, you just say the inverse square law. You don't need to know exactly what an inverse square law is, but if you're doing maths, it might be something that you want to look up a little bit more. But it's the inverse square law. As distance increases, the light intensity decreases, but the proportional relationship is between the inverse square of distance and light intensity. Now, of course, it would be useless to talk about photosynthesis without actually talking about why it's useful. And it's useful because it produces glucose. Glucose C6H12O6 is fundamental in all of life. Well, okay, most of life. But life as we know it, the majority of it uses glucose or a derivative of it as a source of energy. So plants take light, they take light's energy, they produce a chemical energy store, which is glucose. Plants then can spread that energy by being consumed, by decomposing into other organisms. And that's what's fueling all of life. You know, we would not be here, not be able to do this thinking about photosynthesis were it not for energy that we get because of photosynthesis. So this energy source, this store of energy can be used for respiration. That's in plants too. So plants and animals will use respiration. The glucose is combined with oxygen to produce energy and water and CO2. So that's number one. Plants will do that themselves. They have to make glucose so that they can use the glucose. They're converting light energy into chemical energy. They also can use it to produce storage. So they can produce fat or oil. And those fats and oils, just like we produce fats, is a storage of energy. It's a really dense way of storing energy. It's locking it away until it's needed. They can also use it to produce cellulose. Cellulose is a type of carbohydrate. We can't get energy from it, but plants use it to produce cell walls. It's what makes them strong. And they can also use them to make amino acids. And amino acids are used in protein synthesis. So many amino acids make a protein. So really, we're talking about here the production of the three main biological molecules, the three ones that you would have looked at in digestion, of carbohydrates, of lipids, which are the fats and oils, and of proteins from amino acids. So glucose is the fundamental ingredient in all of these molecules. So glucose is absolutely essential, which is why it's so important to learn about what photosynthesis is and how it works. But thank you for watching, guys. Hopefully that's been useful. Do like and subscribe. Click the bell if you want to check out for when more videos come out. And if you have any questions, do put them in the comments, or any suggestions, put them in the comments.